Today, we're going to talk about a piece of technology that honestly sounds like it's straight out of a science fiction movie. But it's not. It's happening right now in labs all over the globe. We're talking about a tool that could let us rewrite the very DNA of our food, and in the process, maybe solve some of the biggest challenges humanity has ever faced. So yeah, let's get into it. But first, you might be asking, why? Why on earth do we need to rewrite our food? Well, the simple and pretty alarming answer is that we are staring down the barrel of a global food crisis. And the stakes, well, they couldn't be higher. And to really bring this home, let me just ask you this. Can you imagine a world without chocolate or oranges? I know, it sounds totally dramatic, but some scientists are warning that climate change could make chocolate a real luxury item in as little as 40 years. And right now, citrus crops are fighting for their lives against a devastating disease. These foods we love, they're really just the tip of the iceberg of a much, much bigger problem.
And that problem is all about scale. See this number? By the year 2050, the world's population is expected to hit 10 billion people. 10 billion. To feed everyone, experts say we need to increase global food production by 100%. We need to double what we're making right now.
And making things even harder is the fact that our crops are basically fighting a war on multiple fronts. You've got biotic stressors, things like pests, fungi, bacteria. Then you have abiotic stressors, like drought and crazy heat waves. All of this is getting cranked up to 11 by climate change, and oh, by the way, we have less farmable land and less fresh water to work with every year. The result? We're getting smaller harvests right when we need them to be bigger than ever. So the challenge is absolutely immense. But what if science has an answer? What if there's a revolutionary new tool that could give our crops a real fighting chance? Well, there is. It's called CRISPR. So what is it? Put simply, CRISPR is a technology that lets scientists act like genetic surgeons. They can go right into a plant's DNA and make incredibly precise edits. Maybe delete a gene that makes it vulnerable to disease, or boost a gene that helps it survive a drought.
You know, the best way to understand CRISPR is with an analogy you already use every day. If you've ever used find and replace in a document to fix a typo, you basically get the idea. CRISPR finds a very specific sequence of DNA letters, and then it can change them. It's that precise.
So how does this genetic find and replace actually work? Well, you can break it down into 4 main steps. First, a little guide molecule zeroes in on the exact gene that scientists want to change. Second, a protein, acting like a pair of molecular scissors, makes a clean cut in the DNA right there. Third, the plant cell's own natural repair crew, which is amazing by the way, rushes in to fix that break. And fourth, as the cell makes that repair, the new, desired edit gets locked into place. Simple as that.
And this isn't just a cool idea on a whiteboard. This process is already being used to make real, tangible changes to the food that might end up on our plates one day. The potential here is just astounding.
I mean, just look at this. This is a taste of the future that's already in the works. Scientists are using CRISPR to make wheat that's resistant to a devastating fungus called powdery mildew. They're editing corn to make it more tolerant to drought. They're tweaking tomatoes so they have a longer shelf life, which could slash food waste. They are even working on those oranges we mentioned, making them resistant to citrus canker. And get this, they're engineering cassava, a staple food for millions, to be cyanide-free.
So the promise is undeniable, right? More resilient, more nutritious, maybe even safer food. But let's be real, a tool this powerful is always going to spark some debate. And CRISPR is absolutely no exception.
And this brings us to the big questions, the ones you're probably thinking right now. Is this just another GMO? And the even bigger question, is it safe? These are the really crucial points at the very heart of the global conversation happening around CRISPR. And here's the thing. The world is totally divided on how to answer those questions. In the United States, the approach is basically product-focused. They look at the final crop. If the genetic tweak made with CRISPR is something that could have happened naturally through traditional breeding, it generally faces fewer regulations. But the European Union, they take a process-focused approach. They regulate the technology itself, and right now they consider most gene-edited crops to be GMOs, which puts them under super strict rules. So, let's talk about safety. The main concern often boils down to this. What if those molecular scissors mess up and cut the DNA in the wrong spot? That's called an off-target edit. And look, it's a totally valid concern. But the research so far shows that in plants, these kinds of mistakes are pretty uncommon. And there's a huge safeguard built into the process. Scientists screen thousands of these edited plants, and they painstakingly select only the ones that have the perfect edit and absolutely no unwanted side effects. So all of this brings us to the future. With the science moving at lightning speed, the biggest questions we have to answer might not be technical anymore. They might be social.
As one recent scientific review put it, the biggest roadblocks for CRISPR-edited foods, they're not really in the lab anymore. The hurdles are in our laws and in public opinion. Building that trust with the public and with regulators, that is the next great challenge.
And make no mistake, this future is coming up fast. Some experts estimate that we could be seeing the first CRISPR-edited foods on supermarket shelves within the next 5 to 10 years. This is not some distant, abstract idea. It's right around the corner.
So, we're left with this incredibly powerful and kind of provocative question. For the first time in human history, we have a tool that lets us directly and precisely rewrite the genetic code of the things we eat. The science has given us this incredible power. The real question for all of us now is, how are we going to choose to use it?